mind if you can hear me okay? Perfect. Thanks, Charles. All right. So we have two different stories we're going to talk about today. The first one is a kind of gene regulatory network story. It's published. It came out earlier this year. So I'll go through that quickly, but then you're the first set of people who are going to hear the story that has a provocative title, which is mechanisms of direct repression by a transcriptional activator. Okay. All right. So we study gene regulation, specifically mechanisms of transcription here. We'll talk a little bit about protein degradation today. So I want to briefly talk about two of my favorite Nobel Prizes because they do two really cool things. They're question-driven science and they're really well-designed screens. And we're genomicists, but we always start our projects with a question as opposed to generating a bunch of data and then identifying the question. One of my favorite Nobel prizes is from '95, and this is in the 80s when people were taking Drosophila, which I agree, I don't study Drosophila anymore, but I do agree that it is the best model organism and the best organism. And it's, I had a few land on my hand today, and it was kind of brought back some memories when I was in your office as well. Anyways, these Kristani Nassan-Vollard, Eric Weishaus, and Ed Lewis, they took male Drosophila and mutagenized them, and their question was, what genes are important for developmental patterning? And they found a bunch of different genes. So here's an example of a gene that had a mutation. So these are pseudo-colored flies. The mutations aren't the colors. But you can see this fly here has an extra thorax and an extra set of wings here. Here's another really provocative phenotype where you have legs growing out where the antennae are supposed to be. So they did these classic genetics, and they did the mapping, and over half of the genes that they found encoded transcription factors that are important for this developmental patterning. And even the genes here that they found, particularly in the cell signal receptors, kind of converge on disrupting transcription factor function. So I like classic genetics. Erica was telling me she teaches the classic genetics class, and that's the class that really got me excited about biology and doing research. More contemporary Nobel Prizes from Shinya Yamanaka, who shared the prize with John Gurdon. And what they did was they looked for, using microarrays, genes that were more highly expressed in pluripotent stem cells versus differentiated cells. And they got a bunch of candidate genes and they kind of brute force co-express these in differentiated cells and asked, what combinations of factors are important for coaxing these differentiated cells to become pluripotent. So the 4 factors that they found were all encoded transcription factors. There were other non-transcription factor proteins that were present in the screen, but these are the ones that drive differentiated cells to become pluripotent. And if you want to differentiate iPS cells, you'll turn on different transcription factors to get a muscle or a fat cell or a neuron or a red blood cell. Okay, so transcription factors are important. So what are the questions that drive our research program? I'm going to introduce you all to this kind of new flavor of gene regulatory networks where we have 2 different nodes. So this is a bipartite graph. You have regulatory elements in a genome. You have genes. If you have an arc or an edge between a regulatory element and a gene, it means that binding of transcription factors to this site is regulating this gene. If you have an edge from a gene to a regulatory element, that means 2 things. It means that the gene encoded a transcription factor, and we're inferring binding to all these sites in the genome. So if you regulate a gene that's not a transcription factor, that's a terminal node, and you can think about this cascade, propagating forward, right? So you have different waves of binding of transcription factors, activating different genes, and our focus really is on the transcription factors. And the question we're asking are what transcription factors are important in these regulatory cascades. Sometimes they'll be transiently activated or repressed, and that will be important. And our idea is to leverage the kinetics of these cascades in order to identify new transcription factors that were not previously shown to be important. So some approaches, we could use ChIP-seq to figure out where all the transcription factors are binding in the cascade, but really we need to kind of know which transcription factors to focus on. We could do a TAC-seq. That gives us the putative regulatory elements, but then we have to figure out which transcription factors are binding and dissociating during this cascade. We can do RNA-seq, that measures changes in gene expression. But if we're looking at really fast time points, like we have to wait for RNAs to turn over to detect repression and to accumulate over background to detect activation. So we might want to do a different nascent RNA sequencing methodology. This is a question that we had when we started out that we didn't have an answer for. We had to empirically determine what time points we'll choose for looking at these differentiation cascades. And we wanted to choose a model that we could genetically manipulate to validate factors, right? But we don't want to just generate a gene regulatory network. We want to be a genetically tractable model where we can knock out a factor and see what happens. Okay, so as Erica mentioned, I kind of jumped between like organisms like Drosophila and in humans and different hormone signaling. So with this, well, I guess I'm getting ahead of myself, but we use a dipogenesis to study that. Another question that we have in the lab that's really been driving my my research since a graduate student are what steps, you can't really see over here, what steps in transcription do each of these transcription factors regulate? So a lot of stuff has to occur in order to go from closed compact chromatin to getting productive RNA, like chromatin decondensation, polymerase recruitment, initiation, pausing, as Erica mentioned, pause release, and elongation. And what I want you to focus on here is like all these enzymes, polymerase, NERF, GTFs, NELF, like they don't know where to go in the genome, right? So every gene essentially needs to have all these factors go here, but they're directed by these sequence-specific transcription factors that bind to DNA with a specific sequence, and they have an effector domain that preferentially interacts with cofactors. And these cofactors tend to specialize. Likewise, the transcription factors tend to specialize. So we're interested in like how in classifying transcription factors, not just as activators or repressors, but like what step in transcription do they regulate. So we'll get at both of these. So the way we think about transcription is we have like a bunch of motifs that sequence-specific transcription factors bind to. They all kind of specialize in the cofactors that they recruit. Some transcription factors are more selective and some are more promiscuous than others. And together they kind of coordinate or they antagonize one another to regulate expression. And the way that we study this is we kind of systematically get rid of a factor, in this case this pink factor, and then see how that affects transcription. And we can say using some computational methods whether or not that cofactor regulated chromatin decondensation, initiation, pause, release, or elongation. So, and we always, Erica mentioned that we studied heat shock as a PhD student, and I started studying hormone signaling, and that's because we were really kind of wedded to this idea that we need to rapidly activate or rapidly degrade factors and then kind of track the mechanistic events that follow. Okay, so the first part is this mechanistic gene regulatory networks. So we have these, remember, we have two nodes, regulatory elements and genes, and each of these edges is a mechanistic inference where it's cis activation or binding. Kind of in cartoon form, you have binding of a transcription factor, this regulatory element A, regulation of this gene. If this gene encodes a transcription factor, you have binding to that transcription factor, places in the genome, and then activation of local genes. The model we use is a dipogenesis, not because we're like we had any prior experience in this, but because it's rapidly inducible and 3T3L1 cells can be genetically manipulated. You add 3 drugs, a glucocorticoid, a CAMP agonist, and insulin to these pre-adipocytes, and 6 days later you have an adipocyte. So our experimental design, and I'll talk about what ATAC-seq and PRO-seq are, but you take these cells at time point 0, you do ATAC-seq 3 replicates and PRO-seq 3 replicates, and then the time course is every 20 minutes for the first hour, and then every hour until hour 4. So we have 7 time points in a really short period of time. And I'm not going to talk about it. We also did a 6-day time point. And the reason we did this is because people have been studying this for decades, and the earliest time point that people have looked at had been 4 hours, and we wanted that to be our terminal time point with the idea that a lot of stuff's happening in 4 hours, and we didn't really know how much was happening, but okay. So ATAC-seq measures chromatin accessibility. There's a lot of different ways chromatin can become accessible, but it's just a general measure of chromatin structure. And what's important for us is we can look for peaks in the genome, and you can look for overrepresented motifs in these peaks, and you can infer transcription factor binding. Of course, you'll want to test that. Basic idea is if you have your DNA wrapped around nucleosomes and your transcription factors binding here and coordinating kind of an open or nucleosome depleted region, you can get cutting, and you can look for peaks throughout the genome. And the other method we used, and Erica was one of the first people to use this kind of genome-wide run-on assay to detect NAICS in RNA, this is called ProSeq. It was developed by Hoo-Jun Kwok and John Liss's group about a decade ago. And the idea is that if this is your transcriptionally engaged RNA polymerase, and this is your RNA here, you isolate nuclei, you wash out all the NTPs, and then you do a nuclear run-on assay with NTPs that can be affinity purified. In this case, they're biotinylated. And these are actually functionally terminating, where the polymerase can't really incorporate more than one or two of these into the active site. So you have really high spatial resolution of where polymerase was transcribing, and you know the direction that it's going. So you cut up the DNA, and you do it a library prep, and you sequence from the 3' end, so you figure out where polymerase was. And the cool thing about this is if condition 1 and 2 are like separated on the order of minutes, you can still detect activation and repression, whereas in RNA-seq, you would actually have to wait for RNAs to either be turned over to detect repression or to accumulate over the existing levels to detect activation. What's different about RNA-seq data is like you get comparable coverage over introns. So if you want to study splicing, this is probably not the assay for you. And you have this pause peak here. I'll talk about that a bit later, but this is where RNA polymerase transcribes less than 100 nucleotides and kind of pauses and waits for signals to proceed into the gene body. So here's all the data. We have over 200,000 ATAC-seq peaks, 30,000 genes. So here's some peaks, here's some genes. But for the purposes of this talk and this analysis, we're only going to focus on those peaks that change over the first 4 hours in the time course. And we're only going to focus on the genes that change expression over the time course. So it takes this 200,000, kind of distills it down to 30,000, and the genes distills down to about 10,000. So we'll focus first on the ATAC-seq peaks. Each one of these traces, these red traces, and they're transparent, so once they build on top of each other, it becomes more and more red. Each one of these traces is an ATAC-seq peak, and this is the amount of time of adipogenesis differentiation, and these are the number of traces. And we're clustering them by their kinetics. So these ATAC-seq peaks immediately increase accessibility, and they kind of flatten out. These ones are transient. These have a gradual increase over the 4 hours. And the blue ones are also ATAC-seq peaks. They just are decreasing over the time course. And you have these different kinetic classes. So we are showing 5, and that's what's in the paper, but like you can really kind of break these down to any number of classes depending on how many clusters you want to use. So the next step was to take these clusters and ask, okay, so what motifs are overrepresented specifically in these set of ATAC-seq peaks versus the others? So when we do de novo motif analysis, we get out these motifs. Anyone know what these ones are? Everyone sees this one. So this is AP1, and this is a glucocorticoid receptor. And this is great because we actually added a glucocorticoid, and we added a CAMP agonist that we know activates AP1. So these are good positive controls. It's always good to have positive controls in your coming out of your analyses. But a lot of families came out besides just GR and AP1. Some of them were known to have a role in adipogenesis, and some of them were novel. I'm not going to get into all of these, but I'm going to focus on these 6 here. So there are 14 different transcription factor motifs. These 6 were the ones we're going to focus on because not only is their motif specifically enriched in ATAC-seq peaks that increase accessibility or decrease accessibility, so not both. The expression of transcription factors that bind to these motifs are also changing. So we're really dependent upon this kind of variation in expression, in the covariation in expression and changes in accessibility to infer these kind of links. And what you want to take from this is like, just because you have a motif doesn't mean you know what's binding to it. Here's an example where this is a motif, and there are 4 different nuclear receptors: the androgen receptor, the glucocorticoid receptor, the progesterone receptor, and the mineralocorticoid receptor. They all bind to this motif. It's essentially identical. Just because you match to a database and it says like MR or AR doesn't mean that that's what it is. So this is just to be, to just be careful when you're matching the databases. So we can do some clever things like, ask if these are expressed and whether or not they change over the time course to figure out what factors are likely binding to these motifs and driving the changes in chromatin accessibility. So the blueness here is just evolutionary conservation, but the basic idea is that all these family members bind to this motif, all these family members bind to this motif, and so on. We can first categorize these as, are they associated as the motif and the factor, the factor, the families of factors, are they associated with increasing or decreasing chromatin accessibility? And we just ask for all the ataxic peaks, how far away do you have to go to find a CVP motif or a twist motif? And we plot that kind of motif density. It's weighted based on the conformity to the consensus sequence. So some of them are really clear, like twist is associated with decreased accessibility. So it's SP, GR is associated with increased accessibility. A couple of, like, KLF is ambiguous. We know that actually KLF is associated with increasing accessibility, but that's it's a big analysis that's in the supplemental. Another way to ask this is to say, okay, so for all the ataxic peaks that exclusively have a CVP motif or a twist motif or an SP motif, what happens to their signal over the time course? And for things like twist, it's really clear, right? Like, so the vast majority of the ataxic peaks that only have a twist motif decrease accessibility. So we think it's associated with decreased accessibility. And it so happens that these 374 that also have a twist motif conform less well to the twist consensus. So we can say that these 4 are associated with increasing accessibility and these are associated with decreasing accessibility over the time course. So that's just the attack. So the genes also have a distinct activation and repression kinetics. So here's a transcription factor KLF, which is activated very early and kind of, it's one of these gray traces here. And STAT1 is one of these gray traces here. And you can see the kinetics in the time course as well. This is 0 minutes to 4 hours. You can clearly see repression, you can clearly see activation. So now we want to link, in this kind of mechanistic gene regulatory network, we want to link regulatory elements to genes. Those genes encode transcription factors, we want to link them to their binding sites and propagate this forward. So we look for, at all the sites that have like this kinetic profile of increasing accessibility and the presence of this motif, we will infer the binding of the factor that binds to this motif to those sites in the genome. If this ataxic peak is nearby a gene and the gene is activated, whereas the ataxic peak increases accessibility, and they're nearby one another, and we can actually empirically determine from the data how far away factors can activate in linear space. I'm not going to get into that, but we can infer a cis-regulatory interaction between that regulatory element and the gene, and this is really dependent upon the kinetics. If X encodes, we'll say, a repressor, we can incorporate a time delay for that repressor to be transcribed and translated, and then bind places in the genome. And if, say, D contains the motif of that transcription factor X binds to, and you have a decrease in accessibility, we can infer binding with that repressor. And say the gene, so this is the pro-seq signal of one of these genes, this gene Z, we can, if they're going in the same direction, they're nearby one another, we can infer a repressive edge between that regulatory element and that gene. Okay, so we did this with all the data, and we're interested in these highly connected transcription factors. So here's the raw data on the right, here's the network. We activate these immediately with agonists for both of these factors, and we can infer binding within minutes to these places in the genome. Activation of this factor called TWIST2, we see an increase a decrease in accessibility of places where TWIST2 binds in the genome, and repression of these factors SP1 and SP3. And we can see, because we're repressing and these are activated, we can see their subsequent dissociation from places in the genome and repression and attenuation of this factor here. So we can see in the first 4 hours, we're pretty confident that we have 3 waves of binding of transcription factors and changes in local transcription of transcription factors that can propagate this forward. Okay, and so a lot happens in 4 hours. So recall that Twist2 here was one of the immediately activated genes. And I think one of the hardest things in kind of network biology is kind of not putting hairballs up on screen. So like I have all these different networks that have very different, so this is supposed to show like how deep the network is for the data. Here is to show how connected Twist2 is. Twist2 binds to thousands of places in the genome, regulates hundreds of genes. The genes that have names next to them are transcription factors, so they can propagate this network forward. Here's SP1, SP3, showed previously, that bind a bunch of places in the genome, regulate a bunch of transcription factors that are also regulated by Twist2, and this can get very complicated very quickly. But the take home from this is that Twist2 is at the top, and there's a lot of genes that are downstream of Twist2. So our hypothesis now is that if we perturb Twist2 and then we undergo adipogenesis, it will affect adipogenesis. So we did a lot of stuff actually in the 3T3L1 cells that's in the paper that I get into. I think what's probably more compelling is just this kind of picture. So we ordered some Twist2 mice from JAX and started phenotyping them, or having histologists phenotype them. I'm not a pathologist here, but so I will try to explain this. So these big white circles are absent over here, and these are fat deposits. So that gave some kind of suggests that in these Twist2 heterozygous mice that they Twist2 could be involved in adipogenesis. Here's the brown adipose tissue and Twist2 minus mice, and there's less white circles in the minus mice. So it is strictly possible that, like, because these are not tissue specific knockouts of Twist2, so it could have a function in some other tissue that's feeding back on the subcutaneous and brown adipose tissue. So we're going to test that as well, and I'll talk about that a little bit in future directions. The other thing we're really interested in is, I'll move this up. Move it down here. I don't know. Let go. Whatever. So what are the molecular functions of these key transcription factors? Oops. All right. Remember, transcription factors tend to specialize in the molecular function. And people have been actually coupling nascent RNA sequencing to transcription factor activation for many years to figure out what transcription factors do. Estrogen. This is work from Lee Krause and John List, where they treated cells with estrogen for 10 minutes, and they showed that ER predominantly regulates Pol2 recruitment or initiation. This is work also from Lee Krause, but with Charles Dinko led it, who's now a faculty at Cornell, showed that TNF alpha regulates elongation. Work from Fabiana Duarte showed that HSF regulates pause release. But these were all one-offs. Like, you treat with estrogen, you treat with heat shock, you treat with TNF alpha, and you regulate, like, or you activate one transcription factor. But now we have a cool system where, like, we're activating 14 families of transcription factors within the first 4 hours, and we can design these, like, this is what I call a GR-centric network, where, like, GR is binding to all these places in the genome, where we have evidence for GR binding to all these places in the genome. Here, there's only evidence of GR binding, and here there's only evidence of GR binding. However, these regulatory elements is evidence of GR binding and some combination of these other factors. What we really want to focus on are the genes where there's only evidence of GR binding upstream to regulate the genes. So now, and we could do this for all the different transcription factors. We can say, GR is the predominant regulator of this 174 genes. And now we can start asking the same question. So what does GR do? Where does it regulate transcription in the transcription cycle? Yeah, absolutely. Yes. So the predominant feature of chromatin that determines whether or not GR will bind in the genome is how accessible it is before treating with dexamethasone. So it has a sequence that it likes to bind to, and how well that sequence conforms to the sequence GR likes to bind to, and how accessible the chromatin, a function of those two variables, determines whether or not GR will bind there. So it binds to a bunch of places. Not everywhere it binds actually causes changes in gene expression. In fact, here is 1,000 places where GR is binding, and we have no evidence that it's changing gene expression. So in fact, transcription factor binding does not mean it correlates to or corresponds to changes in gene expression. Did I answer the question?
Okay. So how do they, why does the different molecules go to different places? Why don't they all want to go to the same?
Well, because I mean, you could, there's a maximal occupancy of what, like, one molecule per site, we'll say. Well, it's just dimer, so it's like 2 molecules per site. Does that answer the question? So like, they can't all go to the same place because like once it's occupied, it can't be occupied by another molecule. Yeah, they do that. I mean, I don't know if it's a decision. I consider it like a mass action, right? Like they become competent to bind to DNA, and then if they have a motif in a region that's open, they will bind. Yeah, so we think of it as mass action mechanistically. All right, so the way to actually figure out, so this is kind of the transcription cycle broken down into a rate for initiation, premature termination, pause release, and elongation. And you can actually think about the density of polymerase in the pause region and the gene body as 2 measures that we get from ProSeq. And we can actually develop this into a compartment model. So time 1 is you have density of polymerase here, and density of polymerase here in the gene body, and time 2 you have these different densities in polymerase. So we can actually develop ordinary differential equation to describe this process here. It's fairly straightforward, and we have empirical measurements of pause density and gene body density from ProSeq data, and we have really good measured rates of elongation from many different labs and many different experiments. And we can just ask, what rates of initiation, premature pause release, and pause release can explain the empirically observed changes in polymerase density in the promoter in the gene body. And we're going to specifically focus on these 174 GR responsive genes. So here's the actual changes of polymerase density in the gene body for the average GR responsive gene. These are the changes in the sorry, this was the pause region, this is the gene body. And we ask, okay, so once we have a set of parameters that can explain the initial data set, how can you change initiation rate and pause release rate, which tend to be the regulated rates in transcription, and account for the observed density changes in the pause region in the gene body. And the only way that we can explain this is by an increase in pause release rate and very modest changes in initiation. We're holding premature termination and elongation constant because those aren't regulated steps typically, and I'm happy to talk about that later. So this data suggests that GR regulates pause release. And we can actually, if we make some assumptions about the elongation rates, we can actually get residency times for GR in the pause region and how they change, and like rates of initiation, like how many molecules per minute, just from ProSeq data, and really good measurements of elongation that don't come from our lab. Okay, so we did this in the adipogenesis system. So we also took acute lymphocytic leukemia cells, and glucocorticoids have been used for since the 40s to actually treat ALL. If everyone's read Emperor of All Maladies, like some of the corticosteroids that were isolated from, like, I don't know if it's pig kidneys or what Sidney Farber put on or gave to these children that had ALL, and it caused their white blood cells to go down. So anyways, we took these ALL cells that are cultured in the cultured system, we treated with dexamethasone for an hour, and we see a very similar thing where an increase in pause release rate. This is actually a much cleaner system because we know we're only activating GR responsive genes. We see an increase in pause release is what can explain the data, and we get comparable residency times and so forth. Okay, so the conclusions for the adipogenesis network is that if you have a kinetic attack data and pro data, you can infer functional interactions between regulatory elements and genes. You can identify highly connected nodes and maybe new unappreciated transcription factors that are important for the developmental cascade. In this case, we actually get rid of TWIST2, and we see an absence of subcutaneous fat. GR regulates pause release, and we actually have what all the other factors regulate as well. So I think these rapidly inducible systems are really key to providing these mechanistic insights. So for the future of TWIST2, we actually, for the past month, we've had a conditional TWIST2 knockout mouse. We're just kind of confirming that The whole body knockout reproduces kind of what we saw, and that the Swiss2 conditional knockout that shouldn't be just that the genetics work and it doesn't disrupt function of Swiss2 without the Cree. So we're going to isolate the effects of Swiss2 by only expressing only knocking out Swiss2 in pre-adipocytes to make sure it's not like some kind of organismal phenotype that's causing the decrease in adipose tissue. And something I'm not going to talk about, but like Twist1 and Twist2 actually have the same binding site. We think they antagonize one another. So I'm just going to take like 2 questions before I go into the next, and I prefer to take it from a graduate student or a postdoc. If anyone has questions, otherwise I'll just take a sip and I'll go into the next. All right, open it up to faculty. Surprising. We didn't do any 3D chromatin conformation changes. Yeah, but yeah, we should. My colleague Jill Doan thinks we should do these kinetic time points with micro C data, and I don't disagree. But I think it's a big cost to do those time points. Yeah, with the chromatin conformation changes. All right, any other questions before I go into the new stuff? All right, so I'm going to talk about this mechanisms of direct cis repression by a transcriptional activator. And just for a background on this, like for the past like 5 years, I hear a lot about, like even in the transcription field, about like this transcription factor activates in this context and represses in this context. Or no, it's an activator, but it's usually said it's an activator and repressor and it's context specific. And I always ask what the context is, and I never really actually get what the context is. Everything that I've studied, so I've studied heat shock factor, ER, GR, AR, like they tend to be activators or repressors. I mean, there are situations that like where we think like if a really crappy activator like competes with a binding site for a really good activator and it looks like a repressor, but the molecular function of it being an activator is kind of like not changing. So I think that the bar for saying that it's an activator and a repressor should be really, really high. But hopefully I'm going to convince you today of an activator whose molecular function doesn't change, but it acts to repress genes. So this is everything. This is my entire career here. So I've only studied rapidly inducible things. And I always say it's because I'm like, it's really hard for me to think about things like 4 hours apart, and so much stuff can happen in 4 hours. I can't get my brain around that. So I don't know how you study development. Don't do it. Cool. Unless you have these high, minute time points, maybe you can think about doing it. So I studied Drosophila or heat shock factor in a lot of different organisms. Yeah, it's not in the nucleus, and you heat shock, it goes in the nucleus. So we can track mechanistic events. I started studying estrogen receptor because I wanted a job. Unfortunately, that job tends to be in medical schools. So I should have stayed with Drosophila. I started studying glucocorticoid receptors when I got my own lab, and a little bit of androgen receptors. But these are factors that we know a lot about because we've been studying these for a long time because they're rapidly inducible. But these other factors that we're interested in, I'm not going to get in too much why we're interested in them, but suffice it to say that we have evidence that these interact with estrogen receptor to regulate gene expression. We're interested in that cooperativity and how they coordinate with one another or antagonize one another to regulate transcription. But we can't rapidly activate or induce these. For instance, like ZNF143 is essential, so you get rid of it, the cells die. We think it's involved in estrogen signaling, and to be honest, we tried to tag a lot of these things with degrons, and this is one of the first ones that we tagged. So I've been using RNAi actually my whole career as well, and knockouts to study molecular mechanism, but there's problems with this, right? Like if you deplete something for the lifetime of an organism or 3 days, like you get feedback, right? You're not even looking at the primary effect, you're looking at the post-secondary effect, and that can dominate in terms of your phenotype. Worst case scenario, you're phenotyping dead things. So degron tagging is a real attractive alternative, and I started oxygen-inducible degrons when I started my lab. So I used to be a mechanic, John taught me how to like take any kind of molecular mechanism and make some obscure metaphor. So stick with me here. He thinks of everyday phenomenon that is like molecular mechanism. So, all right. So, what does the car's radiator do? If I want to use RNAi to study this, I will kind of poke small holes in the radiator, and I will wait 3 days. And so these are my omics that I'm doing here. These are my like gauges. So I wait 3 days, and at some point on day 3, like something happens, but I'm not looking at my cells at that point. And I'm going to phenotype these all. If you notice, when the car was on fire, the temperature was like really high. So I look at my gauges on day 4, and my conclusion is that the radiator keeps the car healthy, right? It affects all this stuff. So what we want to do is want to be driving like 60 miles an hour and just like rip out the radiator, and then monitor these gauges here while we rip the radiator out. And you monitor, you phenotype it like continually. We'll do like a time course, and we see, okay, here, so rip out the radiator and temperature goes up. So it directly regulates temperature, and then like all the other effects on like the starter and all this stuff is indirect effect of the car catching fire. So this is my pitch for using degrons instead of RNAi. Okay, now we know what a car's radiator does. All right, so you tag your favorite protein with this FKBP12 variant. This is a D-tag system. Basic idea is you add a small molecule. The molecule doesn't do very much except the protein that you have tagged. Assuming tagging doesn't disrupt the function of your protein, you can add this molecule and it degrades your protein very rapidly. It shuttles it to the proteasome. So now we can start taking transcription factors, degron tagging them, and seeing, looking at their function. So here's an example of tagging this ZNF143 factor. So here's the untagged progenitor cell line. When you tag it, we have a little bit decrease in expression. So if people don't show you the untagged progenitor, they're lying to you because you always have degradation. This plot would look so beautiful if I didn't show this lane, right? Like it's like, oh look, it degrades within 15, 30 minutes. But in the aid system and the DTAG system, we usually have this kind of background level of degradation. Good thing is we can still study molecular function. So here's a quantitative, standard curve, so we can quantify the degradation. This band right here is actually ZNF143. And you can see by 15 minutes, it's almost gone. 30 minutes, it's essentially gone. So these are just kind of background bands that are unaffected. We don't even think there's ZNF143. So all the degradation data I'm going to show you in this presentation are after 30 minutes of degrading this kind of obscure transcription factor, ZNF143. First thing we did was a ChIP-seq to see if it's off chromatin. This isn't very believable. So these are all the ZNF143 binding sites without the small molecule that degrades it. And you can see, they're ordered by intensity. And 30 minutes of degradation just looks like I put a black box there, but I didn't. So note the scale goes up to like 300 here. If you overexpose this heat map, you can still actually see very small amounts of ZNF143 binding. Okay, but so it's essentially off chromatin at 30 minutes. It has 7 zinc fingers. So this is, just circle them all here. So it binds to DNA. So what motif does it bind to? It actually binds to a really wide motif and it's somewhat degenerate. For those of you who aren't familiar, probably you're in Erica's department, so you look at these motifs more than you'd like, but this is a very wide motif. And for just to give some context, like a letter height of, this height is 96% of the times when ZNF143 binds, it's a G at this position, and 61% you have a T at this position, and so on. This is, compared to the genome-wide background. And this is not inconsistent with something that has 7 zinc fingers binding a very wide degenerate motif. So you have degeneracy that's tolerated in more positions compared to others, because you have these fingers that can come on and off. Okay, so we degraded it really rapidly. Here's the chip. You can see the motif's essentially gone. And you can look at peaks that change accessibility as well. So each one of these dots is an attack-seq peak. And we're looking at what happens on the y-axis between no degradation of ZNF143 and degradation of ZNF143. So a dot that is below X or below 0 on the y-axis is decreasing accessibility. These are controls that we compare to. So if you're I just, for all of those people, we're in a data science institute and a computational molecular biology group. It's really important to not compare your activated and repressed, or in this case, increasing accessibility and decreasing accessibility, to a set of regions that are expressed at similar levels or have similar accessibility. Because like if you include like all these gray points, like they're just inherently different. Like they're not controlled for the levels of accessibility. So anyway, so these are all the the ataxic peaks that decrease accessibility. For example, this one right here is actually the peak that I showed you. And we can ask for all those that are decreasing accessibility or increasing accessibility, what fraction overlaps ZNF 143 binding sites, and it's nearly all of them that decrease accessibility. And you can see none of the ones that increase. So this is saying that like the 30-minute effect is a primary effect. ZNF 143 is probably decreasing the accessibility of all these sites to the genome, and these might just be, I don't know what they are. There's only 4 of them. So then we're going to use ProSeq to quantify changes in gene expression. Unlike ataxic peak, we have a bunch of genes that go up and a bunch of genes that go down. And I'm going to use the up and down nomenclature because we're degrading something. So if it goes up, that means like it was originally acting as a repressor, right? And we're degrading it. And so I'm just going to use up and down after ZNF 143 degradation. So a bunch of genes that go up, a bunch of genes that go down. I'm going to explain how we interpret like cumulative distribution functions. So we have a bunch of genes. So here's the genes that are maybe unchanged here. These go up, these go down. I think I changed the nomenclature, unfortunately. So these are up and these are down. For all these genes, so to say there's 4 genes that's unchanged, we ask, or so for the activated, so there's 4 genes that's activated. How far away do we have to go to find a ZNF 143 binding site? And we go this far away, and on the CDF plot, we do a step of 1. And then the second gene is right here. So this is how we like do a CD ask. So for this set of genes, how far away do you have to go to find the first ZNF 143 binding site? And if these things are shifted to the left, that means that ZNF 143 is really close to those sites. So in these repressed genes, for instance, they're no closer to ZNF 143 or this pink transcription factor than unchanged genes. Okay, so a left shift indicates that the activated genes tend to be closer to the pink binding sites. So this is for every factor I've ever looked at. Like, and this is why it convinced me, like at least before I started this project, that like the activators be activating, repressors repress, right? So because for a bunch of genes that are activated by estrogen receptor, and a bunch of genes that are repressed, only the activated genes are closer to ER. The other ones are not any closer to estrogen receptor than random set of genes. And this is for every transcription factor that I've studied in this way. So HSF, ER, GR, and AR, not both. So when I started doing this for ZNF 143, kind of changed my worldview because like 94% of the genes that are down are near ZNF 143. In fact, within 440 bases. But the genes that are up are closer, like statistically, like I couldn't ignore it. I was convinced they were closer to ZNF 143 binding sites than a random set of matched controlled genes. So I really had to like, how can an activator be repressing? And I'm saying it's an activator, remember, because it's really close. So those that decrease accessibility when you degrade it tend to have the ZNF 143 binding site, and those that are down when you degrade it tend to be, all of them essentially are close to ZNF 143 binding. So how's it doing both things? So these are closer. So we think these are like happening in cis, right? And the way that we determine this kind of threshold for being in cis is we ask, when do these curves kind of converge, become parallel with the tangent of these curves become parallel with the control match set of genes. Okay, so how does ZNF 143 activate gene targets? So the first thing we did is we kind of looked at the genes. So these are all the genes that go down. ZNF 143 clearly binds in the promoter of these genes to activate them. And some of these actually, where we have strong binding downstream of the promoter, it's just poor annotations of the TSSs. What's weird about the genes that are, don't conform to my worldview is they tend to be upstream, over the TSS, and downstream. So we think that normally ZNF 143 binds in the promoter to activate genes. So that's how it normally acts. And we actually took all these genes that are activated, they go down, and we plugged them into the compartment model. And it was very clear that, so each one of these is each one of these dots is a gene. And we did the compartment modeling on all the different genes, and in almost every case, a decrease in initiation rate could explain the data. And pause release actually tends to go up slightly, but there's no clear shift one way or another about pause release rate. So we think ZNF 143 regulates initiation. Okay, so how does it repress target genes? Okay, the first one was really easy. I was like, okay, these things bind over the transcription start site. How does that work? So, Erica, you might like the genes I picked. I actually picked genes, like the target genes that are actually important in transcription. But so here's a ZNF143 motif. Here's a ZNF143 binding site. And when you degrade ZNF143, we actually see more. So this is genes transcribed from left to, or sorry, from right to left. We actually just see more transcription. So what we think is it's just occluding the TSS. We don't think its molecular function is changing. We just think it's like binding there and including the TSS, and that's for 5 genes. So I put this in there because the typical like functional experiment that I would do, I would rapidly degrade ZNF143 and see what happens to the expression of this gene. That's how I came to the hypothesis. So I actually don't know what to do to functionally validate this. Right? Like the functional validation was what brought me to this conclusion. Okay, but we did ask. So we did have some expectation for so. There is ZNF143 motifs and binding sites over genes that are matched to up and down-regulated genes, but we had some expectation that they would be stronger sites if they were acting to occlude initiation, and that's what we see. We see the sites where ZNF143 tends to be repressive is binding much stronger than control sets of binding sites. So it's always important to think of binding events not as a binary, as bound or unbound, but as a continuum. Okay, so what we think is happening at a subset of these is ZNF143 binds over the promoter, you degrade it, and you can get more initiation. So it doesn't seem that ZNF143 is actually doing it. It's not functioning to initiate this gene. It's actually just occluding initiation. So the next, we looked at those that are downstream of TSS. Here's an example of the strongest ZNF143 binding site. There's actually 4 ZNF143 binding sites here. We think that it's just acting as a roadblock. Like you get polymerase initiating here and going through, and it has to get through like 4 ZNF143 binding sites, and it just is not capable of doing that very well. And we see for those that are these 28 genes where you have binding downstream, compared to the matched controls, binding is much stronger in those genes where ZNF143 is binding downstream and kind of acting as a repressor. Okay, I just wanted to mention that there are a lot of ways to get accessible chromatin. Like you can have moving around of nucleosomes, changing the acetylation state of histone tails. But one hallmark of regulatory regions and promoters is this bidirectional transcription. So a transcription factor binds and sets up polymerases going both ways. And we actually find that in many of these sites, these 28 sites where ZNF143 is binding in the gene body, that you have bidirectional transcription occurring. So it seems like ZNF143 is doing its job of setting up initiation in 2 directions. But when you degrade it, the bidirectional transcription decreases and you have an increase in expression of the target gene. So it's like polymerase setting up going the wrong way is like inhibitory to expression. So here's an example. So you degrade ZNF143 and these blue and the red peaks kind of go down. So we think not only the binding of the factor, but like if you're setting up pre-initiation complexes and polymerases going the wrong way, like that's going to disrupt transcription and cause repression. So not only the binding is the roadblock, but setting up aberrant transcription in the gene body could act as a roadblock. So I took all those genes. So these are all the 28 genes that have ZNF143 binding downstream. 8 of them have a decrease in bidirectional transcription when you degrade ZNF143. So and if you take the matched ones where you have ZNF143 binding downstream, but there's no effect on transcription, but the genes are expressed at similar levels, none of those have a significant decrease in bidirectional transcription. Okay, so here's what we think is happening. You have ZNF143 being downstream, degrade it, and that's no longer acting as a roadblock. So what's happening at these upstream bound transcription factors or genes? So I'm not going to tell you how I got here because we looked at all places ZNF143 binds and all changes in bidirectional transcription. But suffice it to say that like ZNF143 overlaps other motifs, specifically SP1 motifs. And what we think is that ZNF143 is doing its thing here, and it's activating transcription of LAMTOR, too, and you degrade it, you have an increase in expression of LAMTOR. We think what's happening is that you are now, you no longer have ZNF143 binding to the site in the genome, and now SP1 can bind, and maybe SP1's a better binder. So this is actually testable. So the chip, I haven't done chip in years, but I'm in the lab teaching people how to do chip, and we're looking for SP1. We're asking the question, does SP1 redistribute? So now, like during the no detag situation, you have very little SP1 binding to this site, or in all the other sites, in all the sites where we think there's overlap. And do you see an increase in SP1 binding when you degrade ZNF143? And we would have to just come to the conclusion, if we see that redistribution, that SP1 is a better activator. Here's another example where you have the ZNF143 motif overlapping an SP1 motif upstream of mediator. So this happens at 11 of the 24 upregulated genes. We also see overlap with other motifs like NFY. So we think's happening is ZNF143 is doing its thing. It's being it's regulating initiation, but not as well as SP1, but it's occluding SP1 binding site. And then SP1 comes in when you degrade it, and it does a better job of activating the gene. So of all the genes, this is a distribution of those that are downstream over the TSS and upstream. We think strong binding of ZNF143 could be occurring at downstream at the downstream sites, and sometimes causing changes in bidirectional transcription that are causing bidirectional transcription that acts as a roadblock. Blocking sites of initiation is pretty straightforward. We think factor competition could also explain why it's binding upstream like it's supposed to, but acting as a functional repressor, like when you look at the data, but the molecular mechanism is like not changing, still affecting initiation. Okay, so ZNF143 conclusions are, we think it just binds DNA and stimulates initiation. And even when it represses genes, it's not changing its molecular function. And in this ChIP-seq, if I was talking next week or probably 2 weeks, it takes me a long time to get into the lab, I will be able to tell you whether or not SP1 redistributes. Okay, and the first project was from my first PhD student, Arun Dutta at UVA, who is now back in medical school. And the ZNF143 project is from Jin Hong in the group. And Rujdeep's working on the compartment modeling and actually developing the compartment model that I developed in R into a format that people will actually want to use, and it runs quickly. You can do it on all genes. Tommy, so you say estrogen response and degradation of a different transcription factor. CU degrades another transcription factor that's involved in the estrogen response as well. So maybe 3 or 4 years we'll be back talking about those. Okay, thank you. Mm-hmm. Yes. Yeah. No, I thought about this. Like, is this a conserved regulatory mechanism, or is it just like we're looking at changes in a lot of genes, like we're going to see some garbage here and there? I don't know the answer to that question, but I think like one of them, the one that has like 4 binding sites downstream, that's like a paralog of ZNF143. So I think that's probably conserved. I don't know, but I don't know. I can't imagine that in every case. It's maybe it is, but yeah, that's a very good question. So I mean, that's actually really trivial to do. We can just take those ZNF143 motifs and look at the evolutionary conservation by FastCons or 5LP, or what would you suggest? Yeah, same thing. Yep. So yeah, that's a trivial thing that we'll do. Yeah, we actually do that pretty routinely. Yes.
So, I guess I had a related question.
I think humans all have the same genome, at least when I do science. So no, I'm not a human geneticist. this is one cell type. I mean, I can tell you. So I work and have a lot of close friends who are colleagues, and I give them a hard time. So they'll do an MSA to confirm that their SNP affects binding the transcription factor. I can just look at the sequence and the position weight matrix and tell you whether or not that's going to affect transcription factor binding. So I can, if there are SNPs and you take like a really well conserved G in the binding site and make it a C, it's going to change. But like I actually did, these are HEK cells, so I don't know what the variants are in humans. But yeah, I'm sure there's going to be SNPs in these transcription factor binding sites just like others, and if they don't conform, if they conform more well to the consensus, there's going to be better binding versus the other. Yeah.
Transcription sites where accept the annotation, and the DNF 143 site is in between the two transcription start sites. Does degrading DNF 143 affect what proportion of the two transcripts you get transcribed?
Yeah, that's a beautiful question. I mean, we asked that question for other reasons, but like we could not, in the compartment model, we can't say that something affects initiation and not premature pause release because they're just like opposing like rates. So we had the question like, okay, so if DNF 143 is affecting initiation, the site of initiation will change, whereas if it's affecting premature pause release, it won't change. So we asked that question for a different reason, and the answer is that affects initiation. The predominant TSS does change. It's noteworthy that like we don't actually trust any TSS annotations because like so initiation is actually very dirty, like generally. Like you have predominant initiation sites, but like there's initiation windows, and like at least in humans, like Drosophila has like a bunch of promoter elements, so it's more focused. But in humans, like it's a dirty process. So I don't know where I'm going with this. So we use we have proseq data, and I talked about the 3' end of the data saying where polymerase is. We actually use 5' ends of the data, which is we read pileups at the 5' end, and we use those data to infer TSSs from the data, because the TSS could be different in different cell types and different tissues and stuff. So we don't just rely on annotations, that's our starting point, but we use these kind of read pileups near already existing annotations to look for TSSs, and we do look for changes when we degrade DNF 143, and that's actually how we figure out that it does initiation, not premature pause release. That's a great question.
Yes. I have a question about DNF 143. So you're mentioned it's DNA binding that's blocking SP1 or some other factor from binding.
We think,
so do you add back just the DNA binding domain of that protein in a non-degradable form and test whether DNA binding is sufficient to actually rescue the...
Oh, so you're giving me more work. So just SP1 redistributing isn't enough for you. Now you want me to degrade it and then reintroduce DNF 143 that just the DNA binding domain. Yeah, that's I could do that. If I see SP1 redistribution, I'm sending it off and hoping you're not my reviewer. But I do agree that is a better way. Like it's a you see the redistribution, then you rescue it. Yeah.
Second, more general question is sort of about the answer to the second part of your question. Always is combinatorial control. What makes it doing one thing here and another thing there? So I wonder sort of how you just address the fact that these binding sites for the things that you're looking at in your pathways are attached to other binding sites that are next to them and add that complexity to it.
Well, so I don't know exactly what the question is. So we're trying to keep it as like not complex as possible because we just wanted to grade one factor and see what happens, right? So we know it's complex with the ER system. The idea is to like degrade one factor and like see how that factor works in isolation, and then see how degrading that factor affects the estrogen response.
So I'm saying that factor works with factor B in promoter X and factor C in promoter Y.
Yeah. So
like that difference.
We think about that. I mean, I still think it's doing its thing. Like, I don't know that it necessarily. I think if you put it there, it's gonna do a good job of initiation. But sometimes, if you put it somewhere and it still regulates initiation, it might not do anything to the local gene, assuming you induce binding of it, because that gene isn't rate limited at initiation. It's rate limited at pause release. That's why that gets to like a bigger question is like, I think we need to start characterizing factors by their molecular function, and then we can actually start making predictive models of how factors work together to regulate a profile. So not just say activator or repressor, like this set of activators are regulating pause release, and this is their relative, in relative scale, like how good they are at it. These factors are good at regulating initiation. This is in relative scale how good are they. And then I think all those data together and all the information to have more predictive models of how factors are going to coordinate with one another or antagonize one another to regulate gene expression. I don't know if that answers your question.
I have a sequel question to this question, second question. That is, you alluded to the fact that a transcription factor can be an activator or a repressor. And of course, we know that that sometimes depends on the co-activator or co-repressor. An example would be the ecdysone receptor. When it binds smarter, it's repressed, and then ecdysone comes in and kicks off smarter, and then it's activated. So I wonder, genome-wide, if you look at all of the transcription factor binding sites, are they all either activators or all either repressors, suggesting some global environmental things such as having ecdysone around for the smarter example, or you have examples where in the same cell, in the same genome, some sites will be activators and some will be repressors, suggesting that you may have adjacent binding sites that give specificity.
So I think those are all possibilities, but I think it's important to define the context if you're going to say that, right? So like the ecdysone receptor, I should know about that because of Drosophila, but like thyroid hormone receptor, same thing, right? Like it actually interacts with co-repressors with, it's not a thyroid when there's no thyroid hormone there, but it actually co-activators when you have the thyroid hormone. I consider those different transcription factors, right? Like one is ligand bound and not ligand bound. I think all the not ligand bound are going to interact with co-repressors, and all the ligand bound are going to interact with activators. I cannot conceptualize how it's possible that within one nucleus, well, you make a good point. Like, so say I have a kinase that's maybe like tethered to one place in the genome, and then that can phosphorylate a local transcription factor to change its activity and what it interacts with. So like those kinds of scenarios, I'm open to those. But I think they have to be shown, right? Like, so I think like generally it's going to do its thing. Like if there is a mechanism where like ZNF143 is binding and it happens in be near transcription factor X recruits at a acetyltransferase that acetylates ZNF143 and changes what it interacts with. Like that is a scenario that I'm willing to buy where the cooperating partners or the partners nearby can influence the function of the factor. But like I haven't seen any convincing data for that because it's a hard bar. Like I mean, like the experiments that you have to do are you get to rapid perturbation, like get rid of the factor and like show that the acetyltransferase. Like it's a big bar to go over. So, but I think generally speaking, we're talking about these constitutive transcription factors that aren't hormone receptors. I think they're just activating or repressing, or that's their molecular function. But you can have an activator look like a repressor depending on you have these weird scenarios. I think I don't think the molecular mechanism of the factor acting will be different. I don't think so, but I haven't seen compelling data of that. I mean, but here's an example, like it's a repressor, but it binds over the TSS. So, but like, is it really a repressor or is it like just, binding and that's causing repression? So I guess I'm splitting hairs, but making the distinction between the like molecular function and the consequence of changes in gene expression. I think that's where like we have to, get into the details there to like what we really mean. Hopefully, I don't know if I actually answered a question. Okay. Yeah, I think I'd probably use MNA-seq for that, but like I think you can look at that. Yeah. Yeah. Yeah, like you get a foothold in there, and there's actually several mechanisms I can think of kind of displacing it. Like, I mean, nucleosomes are not, they're not 601 sequence. They're not just like sitting in one place. So they're moving around anyway. So if you have it competing with GR, it's probably GR that he showed. Like, it's maybe not going to go like, you know, it needs accessibility originally, but maybe the nucleosome won't go over there. But like transcription factors going off, nucleosomes going off. So the residency time over that site would probably be less. Yeah, I think, yeah, MNase-seq or attack-seq would certainly allow you to determine that. They're very dynamic systems though, right? Like it's not just like a nucleosome sitting there and the transcription factor binding there. Like the residency times of transcription factors on the order of seconds. And nucleosomes move around a lot in the cell. So thank you so much.
All right.